So before we get started with clicker questions, I had an idea about how we can try and avoid people having to leave immediately after taking their clicker questions and running away. And that is maybe doing online clicker questions and basically just giving people the same period of time that we have in lecture to answer them. You can't get double credit, no. You can't take the clicker questions here and then go back and take them at home and get double credit for those. But just a quick show of hands. I don't know who you are. How many people think this is a good idea? How many people think it's a bad idea? Okay, so we've got a couple things. It's not happening today because I didn't post them, but I'll think about for the rest of the term. So let's try and actually do a clicker question now before this thing actually completely gives up on me. So GTPase activity of which of the following proteins is critical for accurate translocation of the ribosome? EIF2, IF1, IF3, EFTU, or EFG?
Translocation, here we go.
15? 10?
That's what I'm saying. If it was EF2, it's I.
5? Okay. Which of these don't have GTPase activity? Now, that's not the question that I asked. This is review for the exam on Friday. Which ones don't have GTPase activity? IF1 and IF3 do not have GTPase activity. So that eliminates 2 of them. Okay, so now we've got 3 that have GTPase activity. EIF2 is important for what? For initiation. It's for getting the initiator tRNA to the right place, not in the small segment of the ribosome because it's already associated there, but the appropriate place connected to the messenger RNA. So codon-anticodon interactions. So that has nothing to do with translocation. What about EFTU? EFTU, I heard proofreading, which is not really quite what, usually when people talk about proofreading, it's going back and checking that you've actually maybe made the wrong thing and going back and cleaving off the wrong one. This is checking for fidelity, making sure you put the right one in. And so what does that leave us with? G. So EFG, which somehow these guys are on the wrong place. So yes, it is EFG, which is the correct one. And it's, the key here is really translocation. So it's the translocation of the ribosome, the moving of the ribosome across. So our next question, which of the following is most important for the formation of the circular polysome present in most eukaryotic RNAs? Actually, this should be messenger RNAs. Sorry, it should be mRNA there. EF1, 2, 3, 4E, or 4G? Yes, this is, translation is really about alphabet soup. With some numbers, too. Ten. 3, 2, 1. Good. So we all agree on this one. I guess I explained it properly, or you could find it in your notes. EAF4G is the one which is important for interactions between the cap binding complex and the poly A binding protein. What binds to the cap? EAF4E, exactly. So now I select the answers. People who need to leave, adios, hasta la vista. I did have a quick question here at the front about whether I will be posting the clicker questions. Yes, I will be doing that. I can either wait until we have clicker questions on Wednesday, or I could post them today. Today, Wednesday? 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 OK, I'll post them all on Wednesday. So they will be posted there. I've got all of them all together in one PowerPoint file. They're almost ready to be uploaded. But I still need a couple more questions as far as that's concerned. Another couple quick things on the midterm. Again, 50 questions, the nice little blue sheet. Otherwise, it should be pretty similar to the first one. Again, these are going to be lectures after replication, so all DNA repair, translation, RNA processing. Anything I missed? The stuff we're talking about today, which will also be on the midterm. So basically, today, we need to go through this very last step here, the last part of the arrow. We've translated our messenger RNA into a polypeptide chain, but in many cases, that doesn't actually have its three-dimensional structure, which, of course, it needs for function. Oh, slow. Must be Monday morning or something. So yes, in many cases, these polypeptides that are made by the ribosome don't have their final structure, and so they need help. And usually, the help that they need is not doing the wrong thing, which, again, is the whole idea of chaperones, is that they're supposed to be stopping whoever, whatever, from doing the wrong thing. Hopefully, that's what my kids are having today during daycare. School's out. So if, however, they do the wrong thing, the way to deal with that is mostly the proteasome. And so I like to think of this as the other cellular garbage disposal. We talked about the first cellular garbage disposal in the last couple of lectures. What's that? Exosome. The exosome, exactly. And this one is now the proteasome. And again, the molecular biologists love their zomes. And then towards the end of today's lecture, and again, whoever we get is whatever's going to be on the exam. So that's lots of questions. We won't get there. RNA World, talk a little bit about some of the ribozymes and in vitro selection, how you get ribozymes. And this is sort of our artificial time machine going back 3.5 billion years ago, where we think that's at least one possibility for what was going on there. Did ever want to briefly mention these antibiotics, which the table came out really badly on here. But vast majority of antibiotics, I shouldn't say majority of antibiotics, but many antibiotics turn out to be directed at the ribosome. And there are a couple of reasons for this, probably, again, to totally over-anthropomorphize. But if you remember, translation requires huge amounts of energy. And so it's one of those things that you really need to spend a lot of energy on. And so having an antibiotic which directs against the ribosome is going to be a way to really change the energetics of a cell. The other thing, as far as antibiotics are concerned, is that there's a pretty big difference between the bacterial ribosome and the eukaryotic ribosome. And so many antibiotics can function against bacteria, but not against eukaryotes, and vice versa. Although usually we're trying to deal with bacterial infections when we talk about antibiotics here. So tetracycline, streptomycin, chloramphenicol, erythromycin, all of these are specific to bacteria because they bind to the ribosome, and that ribosome is specific to bacteria. There are a few that act on eukaryotes, but not bacteria. These are mostly useful for experimental work. Cyclohexamide is probably the main one. There are a couple more on here that I wanted to mention, alpha-mannitine and rifamycin. These take advantage of the fact that the transcriptional apparatus, so not the translation apparatus, but the transcriptional apparatus, the RNA polymerases are quite different. And so rifamycin works on the RNA polymerase, and alpha-mannitine works on the RNA polymerase. But again, different for the different domains of life. We can get into which ones work on archaea, but I won't get into that in more detail. Yeah?
Is that same sort of mechanism responsible for some of the antifungal type things for use in humans?
So the question here is about, are any of these antibiotics useful for antifungals? Now, antifungals are a really interesting question because fungi are what? They're eukaryotes. And so you can't use any of these broad-spectrum anti-eukaryotic antibiotics without harming our own. And so that's one of the reasons that actually antifungals are a really big problem. And finding good antifungals is a lot more difficult than finding good antibacterials because the molecular basis for fungi and us is really much more similar. Okay, any more questions on this? I'm not going to ask you whether this is, blocking the A site of the ribosome, et cetera. I don't figure that's appropriate. Okay, moving away from these antibiotics, let's talk about the next problem that the polypeptide has, and that is that it needs to have a fully folded structure down here at the bottom to actually have its full activity. But when it's made by the ribosome, it's just a stretch of amino acids. So somehow you've got to get from this stretch of amino acids down here to your final functional protein. And this particular example here, just cartoon-wise, tells you some of the multiple different things that have to happen for most functional proteins in the cell. First, they need to fold up into a particular structure of just the one polypeptide. In many cases, they then have some covalent modifications. And finally, to actually really do their job, they're usually bound to a number of different proteins as well. We're going to mostly be talking about this first step right here. A lot of people talk about unique conformation. You have one amino acid sequence that's going to give you one structure, which will give you one function. That's true in most cases. But again, whenever Stedman puts quotes on his slides, he means that it's not completely true. And the best example is those amyloid proteins that we hopefully talked about. Actually, pre-Dr. Iverson should have talked about before, in terms of proteins which can have two very different structures and have then very different functions, not surprisingly, based on that. But these structures, even if they're multiple structures, are really determined by the primary sequence. And so if you have a particular amino acid sequence, you can get it into its nice stretched out form, either being made directly by the ribosome, or in many cases, people have taken a folded protein, unfolded those proteins, pulled them all apart, and put them back together. You still get the same or the very few number of structures that you started with. So it's really determined by the primary sequence. How do you get, again, to your final structure? First, you usually start with secondary structure. So you get your alpha helices and your beta strands associating with each other. And then in many cases, you get to what's called a molten globule. And that's what's shown here on this next slide. And that is where you have a protein that has most of its secondary structure, but it doesn't quite have its final tertiary structure yet. And it turns out that many, many, many proteins go through these intermediate steps, again, of the molten globule kind of step. And this is where each of these alpha helices, in this particular case, is kind of floating around next to each other. They're not associating. So if you think about coiled coil, leucine zippers, where you've got these amino acids that are going to be associating with each other from multiple different secondary structures. You don't have those formed yet. And also, I like the example here where you've got an alpha helix here, which is just sort of started. This is the molten globule side here. And in the final protein, it's a much longer alpha helix. So you have some changes in secondary structure as well. But very often, this is sort of an intermediate step that these proteins go through. And that's probably because secondary structures form really fast. And it's those tertiary structures which form a lot more slowly. You have a few proteins, and these are often actually the very small proteins. And this is sort of the example that we had from the animation that we looked at last time, is where you have the ribosome making a polypeptide that folds up by itself in the absence of any help, and then this protein is completely folded and active as it comes off the ribosome. That, again, is really very much the exception rather than the rule in terms of how these proteins work. Most of them will go through these molten globule steps. And turns out one molten globule is a very poorly defined structure. So again, it's these secondary structures sort of flopping around relative to each other. So what seems to happen is these secondary structures form, or most of the secondary structures form, and then they basically look for the right other secondary structures to interact with. And again, that's totally over anthropomorphizing. But basically, there's random selection on how these things could be interacting with each other. And basically, what the chaperones do is say, hey, this is wrong. Let's go back over here. Hey, this is wrong. Let's go back over here. And interestingly, at least for me, how these chaperones were found in the first place, they were, of course, found in viruses, because viruses are the coolest things ever. And particularly this one was in virus assembly. And why do I go into this so much? Is I think it helps me at least to understand the terminology, trying to get through some of the alphabet soup. GROW-E. So GROW. GROW is for the growth of lambda bacteriophage. And so again, the geneticists, what do? They break stuff. And then they figure out what they've broken. And so in this case, the breakage, the mutant, GROW-E, lambda can't grow on this particular E. coli. So it turns out that there are two separate GROW-Es, GROW-E and GROW-S, but we'll get back to those in just a second. The other thing here is that many of these proteins were found to be stimulated in terms of their production on a heat shock. And it's not terribly surprising that heating up a cell causes, to some extent, proteins to unfold, but much more importantly, the proteins that are being made not to fold properly. Because you think about heating things up, what does that do? That changes the molecular interaction. So things are bouncing around a lot more. Well, if you're bouncing around a lot more, you're going to end up way more over on this side than you are over here with your finally correctly folded protein. So heating things up does change in terms of what proteins are being, again, not properly folded. And if you don't have proper folding, then you stimulate the production of these chaperone proteins. And so many of them are heat shock proteins. And if you look at their names, they're very often HSP. So HSP proteins, heat shock proteins, important usually for protein folding. There are really two kinds of chaperones. There's the sort of extra protein chaperone. I like to think it sort of sits on the outside of your protein, helps it fold. And then there are the, they call it boxes of infinite dilution chaperones, where basically you put the unfolded protein in a box and shake it up and wait till the right formation happens and then let it back out. So these are the GROW-EL, also known as HSP60 form of the chaperones, where they're basically these big boxes, big on a molecular scale, where you have your incorrectly or incompletely folded protein that comes into this upper container. That then gets closed by a cap. Basically, that gets shaken around, and that shaking around is ATP hydrolysis, where, again, not unlike what happens in GTP hydrolysis for translation, you have conformational changes. And the big problem that most of these proteins have is that there are hydrophobic amino acid side chains that are interacting either with other proteins that they shouldn't be interacting with, or the wrong part of their own protein that's there. And so it's those hydrophobic interactions and reversing those hydrophobic interactions, which seems to be mostly what these chaperones are doing. So here we have hydrophobic protein binding sites that will change with ATP hydrolysis and basically allow the appropriate hydrophobic interactions to take place on your folded protein to eventually get the proper form. Yes?
Does the bottom portion have a function as well?
So the question is, does the bottom portion have a function as well? That is a highly debated topic, whether the bottom part has a function as well. We know in the structure, which is shown down here in the right-hand corner, that there's the bottom and the top actually look pretty similar to each other without this cap at the top, but the cap only seems to form at one end. And so probably it's just happening at one end rather than happening at both ends. But that's a highly debated topic in the field, which one is going on there. There are some other issues with this model, and this is, again, just a model. Works reasonably well in many cases. But if you remember how these things were originally found, GROE-L, GROE-S, lambda growth, putting lambda bacteriophage together, lambda bacteriophage is really big. And there's no way it would fit in one of these containers. So exactly what's going on there, curiously enough, for how these things were found in the first place, is a lot less clear than how they work for small proteins. Yeah?
What happens in an experiment when someone separates them?
What happens in an experiment when someone separates which? The GROE-L and the GROE-S? The two halves. Well, the two halves here, so you mean the top and the bottom? Yeah, so the top and the bottom, these are actually really hard to separate from each other. And they generally form these multimeric complexes, and I'm just going to say multimer because from organism to organism they actually change in terms of how many multimeric states you actually have here. But that is one of the things. But they seem to be really pretty stable. On the other hand, GROE-S, which is this cap structure that comes up here at the top, this pops on and off. And so that you can separate really quite easily. Just separating those two things at the bottom. And so, again, this is a little bit of an aside, but anybody go to the biology seminar Thursday last week? So the fellow who spoke there talked about some chaperone diseases. So it turns out that there are some genetic diseases that only a few people have, but it turns out they're in one of the subunits of this structure down here. So one particular single amino acid change in one copy of heterozygotes can cause massive neurological disease, et cetera. And so there are a few very rare diseases, but in terms of genetic diseases of these chaperone functions. And so those are there. They're clearly very important. Exactly what they're doing is not completely clear. I mentioned, but I'm not sure if I have that as a slide here, there's the extracellular chaperones as well. It's an example in the textbook. But basically, they bind on the outside of these proteins here and block those hydrophobic areas, which otherwise, again, would be associating with the wrong things. Also undergo ATP hydrolysis, and that releases, binds, releases, and binds. And again, basically just seems to provide the opportunity for all the hydrophobic regions on your protein to associate with the proper hydrophobic regions on the rest of your protein. Or, for that matter, sometimes even a different subunit, completely separate polypeptide, where that needs to interact with. So if you think about a newly synthesized polypeptide here, I don't like to call it a protein, newly synthesized polypeptide, some of them get correctly folded without help, and folded is just, again, taking that amino acids, linear strand, folding up into three-dimensional structure. Probably the majority here, and these are not supposed to be really to scale, are correctly folded with a molecular chaperone. Then we have a number of proteins that just don't make it. And these are the ones that end up in the garbage disposal, except for a few. And these are those prion diseases, et cetera. We end up with protein aggregates that can cause a really major problem. Fortunately, that's very few and far between. This doesn't happen very often. What is this protein garbage disposal? Again, we talked about the exosome as being the RNA garbage disposal before. This, not surprisingly, what does it do? Just like the exosome does, it takes the macromolecule and breaks it into smaller subunits. These, of course, is now working on proteins, so it takes amino acids and chops them up into amino acids. Sorry, takes polypeptides and breaks them up into amino acids. In that process, has multiple proteases. And so again, it's one of these structures that, curiously enough, in terms of how it's put together, looks a lot like the GROEL structure. And first people were like, oh, this looks like it's exactly the same thing, but no, they're really quite different. GROEL is helping fold proteins, and the proteasome is helping break down proteins. These are really good proteases, but the main thing here is that these proteases are stuck in the middle of this, again, like a garbage disposal. And this is why a garbage disposal is below your sink. You don't want to be sticking your fingers into it. Things would get chopped up, and you want to control what goes into that particular garbage disposal. And that's what these subunits here are on the ends of the proteasome. 26S, and that's just what people call the proteasome, because a lot of people working on the proteasome well before they actually had sequence data, et cetera. What were they using? They were using ultracentrifuges. So just like S's that you have in the different ribosome subunits, it's the same kind of thing. It's just how big it is. And these different sizes on the proteasome are either with or without these extra pieces on the ends. So the extra pieces on the ends are really the regulatory part. Also, people call them the cap. And they determine what gets into this protein garbage disposal process. How does it get there? It gets there because these proteins have polyubiquitin chains that have been attached in a very specific polyubiquitin chain formation. So we talked about ubiquitination well back, last midterm, where you can have ubiquitination being important for signaling, you can have ubiquitination be important for protein-protein interactions, et cetera. This particular case of ubiquitination is targeting proteins so that they get degraded. This can happen either with what happens with a protein that hasn't gotten properly folded. So there'll be an exposed hydrophobic region. That exposed hydrophobic region then gets these ubiquitins added to it. And it's not just one ubiquitin, it's a chain of ubiquitins which get added. And that then brings this labeled protein to the cap of the proteasome, in which case this now chops off the ubiquitin and basically stuffs the protein into the protease. And not surprisingly, this requires quite a lot of ATPase activity. Is this now, not so much the cleavage of the polypeptide bond, but the unfolding of the protein and putting it down here to get proteolyzed. This is, again, a reminder. It's not just degradation. Proteasomal degradation happens when you have polyubiquitin here leading to degradation through the proteasome. And it's all about the interaction of that cap together with that polyubiquitin chain on the outside. And one of the other things, I need to mention is that because this is sort of what you can think of as a surveillance mechanism, you have to have a lot of the proteasome around. Because if you didn't, then you wouldn't be breaking down these proteins. So it turns out that the proteasome is one of the most common proteins that you find inside the cell. So the most common RNA you find in the cell is ribosomal RNA, exactly. Most common protein is this guy, the proteasome, not these guys, but the proteasome which is there. So again, we mentioned ubiquitination before, histone regulation, this we've already talked about. Cell biology, they'll talk about endocytosis for multiple ubiquitination. Again, these are not made as change, they're just single ubiquitins which are added. Polyubiquitination can lead to proteasomal degradation, but also can lead to other things, including stimulating DNA repair, etc. It's just a reminder how ubiquitination works. On the top here, we have a ubiquitin activating enzyme. This is an E1 enzyme. Basically binds to ubiquitin with ATP hydrolysis and activates this ubiquitin so it can be added to another protein. And the real key here is the E3 protein, because that E3 protein is what gives you the specificity. So E3 tells the E1 what protein it should be putting ubiquitin onto. So it's all about how E3 interacts with things. And so here, an example just down here at the bottom. This is a protein which is going to be ubiquitinated. This signal down here, in this case it's a degradation signal, interacts with E3. That brings the protein to be ubiquitinated to E1 that has the ubiquitin on it. This then gets added. In the case of these proteins which are going to get degraded, it adds this polyubiquitin chain, which is now the signal to the proteasome to bind to this protein and stuff it into the disposal. There are lots of different ways that you can have these degradation signals. The degradation signal could just be a hydrophobic region of a protein that shouldn't be hydrophobic, because usually hydrophobic region should be bound to something else. So you have an incorrectly folded protein. But it turns out that there are lots of other reasons that you want to be degrading proteins. And the classic example is in the cell cycle that, again, everyone in cell biology next term will learn far too much about, or you've already heard too much about it, you're already taking it this term. Classic example here are the cyclins, which you have large amounts of, cause progression through the cell cycle, then you need to get rid of them. We also talked about some of these already. In what context? Very beginning of this section of the course. Eukaryotic replication initiation. So cyclin-dependent kinases, CDTs and CDC6, which are getting degraded after you start replication. So those get degraded due to ubiquitination, that ubiquitination leading them to the proteasome. How does that happen? It happens through phosphorylation. So phosphorylation leads to a change in the structure that now can associate with one of these E3 proteins, leading to protein degradation. These are just a couple of other examples here. One are just protein-protein interactions. So this would be, for instance, a hydrophobic region here on your protein, which is normally interacting with another hydrophobic region. This protein maybe is degraded or it's removed somehow. Now you have an interaction domain for an E3 protein. Or in a couple of cases, and again, these are all examples from cell biology, you'll even have a proteolysis event whereby one domain is removed from your protein, giving you, again, this region that's going to interact with E3. So now we've gone from starting out with our DNA transcription. So this is, again, it's basically a review for the midterm. Transcription, where you start out here, your promoter, you get elongation taking place of your RNA with your RNA polymerase. At least in eukaryotic systems, you have a cap, splice, and tailing which takes place. Once you have capping, splicing, and tailing, now you have export. Once this messenger RNA is exported, now you get translation. After translation, you have to have protein folding. Some cases it's getting degraded, but then you finally have your protein down here at the bottom, which is functional. Yes?
About the chaperones again.
Yeah.
The images, and we were discussing it, looks like a single polypeptide. But obviously proteins are multiple polypeptides. So are these chaperones able to interact with multiple polypeptides at once to create the final shape? Is that what they're
Okay,
doing?
so
Or individual?
yeah, again, I'll paraphrase your question. Basically, the question is, are these chaperones all working together in one big complex, or they seem to be functioning independently of each other? And the answer to that is we don't know. But certainly all the experiments that have been done looking at protein folding have been done on individual proteins, and often on relatively small individual proteins. But you're exactly right, and brings me back to this question about what's going on with assembly of something like a virus, which is humongous. Multiple proteins are having to come together here. So there are probably some different things going on inside the cell relative to what people are seeing in the test tube. And so those are some interesting questions about looking at that. And just recently, we've started to get some techniques where we can look at what's going on inside the cell and now look at where some of these individual proteins are relative to each other. I know the best work has actually been done on the proteasome because that's what you have the most of. But I think people are looking at where these chaperones are inside the cell as well. But it's a great question. Is there another question here? Sue, was that? Sort of, I thought I saw another hand. Okay, so we do have some time to talk about the RNA world. Cool. So this is just a review, or overview, I should say, about the RNA world. We'll talk a little bit about this overall theory on how we think the RNA world may have existed. And again, this is pretty speculative and hand-waving now. We'll talk about how the RNA world could have existed in terms of some of the substrates, what had to have been around if this were to have worked. And the main thing here really is the ribozymes. No, it's not about the viruses, not always about the viruses all the time, but mostly about ribozymes, which you've already talked about. What's sort of the classic ribozyme? The ribosome, exactly. So the classic ribozyme is the ribosome. And it's the ribosome which does what? It makes proteins. And so if you think about some kind of timeline where you started with RNA and then you had RNA plus protein, that would make sense, because what's making protein now? The RNA is making the proteins now. And then again, we may get to viruses. Yeah, we'll probably get to viruses at the end too. Why are people so excited about RNA? Stephen was almost 3 years ago now. Again, talking about this ENCODE project, all the stuff that's not junk that we thought was junk in the human genome to start with. There are probably 20,000-ish RNAs, small RNAs or longer non-coding RNAs that are present in our genome. How does that compare to the number of protein coding genes? Pretty close to the same numbers. So these RNAs, which are clearly not encoding protein, either because they're too small or because they've just been found, and manually curated just means that people have literally found that there are these long non-coding RNAs. Maybe back up a little bit about long non-coding RNAs. What do I mean by long non-coding RNA? Basically, this is an RNA which is as long as a messenger RNA could be, but it doesn't have open reading frames in it. And so that's basically all that you're seeing with a long non-coding RNA. There are no decently long open reading frames in it. What do I mean by open reading frame? Okay, 50 plus amino acids, yes. What else do we need? A start, exactly. You have to have a start as well as 50 plus amino acids that are there. So these long non-coding RNAs either don't have start codons, which is really unlikely because every 256 nucleotides on average you'll have an AUG. But much more importantly, they don't have 50 amino acids or 150 nucleotides without stop codons in them. So these are, again, lots of RNAs which are there. So what about this RNA world? RNA came first. Well, why did people think that RNA came first? Well, if you think about the history of the universe, again, real old history here. 15 billion years ago, plus or minus, quite a few billion years, was the Big Bang. And then between 5 and about 3 billion years ago, life seems to have arisen on Earth, certainly in terms of some of the geologic record. The very, very oldest rocks that you find on Earth, literally over 3 billion years old, have some pretty clear indications of organic material that, as far as we know, can only be formed through living processes. And so that gets a lot to what people are thinking about in terms of how life arose on this planet. And there's also, of course, a lot of interest on, well, are we alone? Is this the only place where life is? And I don't know how many of you have been following some of the astronomy results in the last couple of years, but the number of planets that we've now found around other stars is getting into the thousands, and some of them have pretty similar kinds of, as far as we can tell from this distance, environments to ours. So I think that there's clearly other life out there. It's probably mostly microbial, like most of the life on this planet, but we can continue with that. But a lot of people are interested in thinking about this, and so far it's mostly thinking. Many of them think about this in terms of an RNA world, again, at least what seems to have happened on this planet. Other things may have happened in other places, and again, we can discuss that a little bit more. A lot of this has to do with what people predict the chemistry would have been early on a planet such as ours, and again, some of these origins of life. And so many people think that RNA was a really good candidate for what could have been, and again, just what could have been, the first life. Now, of course, this gets back to how you define life, and life as you know it, and this is a big problem that exobiology and astrobiology have to deal with, is how do you define life? Would you know if you went to another planet and you found something that is really an indication for life? And I think currently, anybody again has been following the Mars rovers, there are good indications that there could have been life there, but none of the sort of smoking gun that there really was life on Mars. So a lot of people talk about various different models, but the main one sort of comes down to this, and that is that at least now, the only molecule that we know of that can both store genetic information but also have catalytic activity is RNA. And sort of the idea here would be that you have an RNA which can catalyze the formation of the same RNA again. And this would be what people also call a self-sustaining Darwinian system, because the other cool thing about RNA is it can undergo mutations, again, because of the genetic material, which presumably could lead to new and different functions. And so this is the idea here of autocatalysis, that you can make some more of that same RNA. So far, in test tubes, we haven't been able to do this, but we also haven't had 3.5 billion years to do it. So this, so far, I think the world record, as it were, of an RNA making RNA, an RNA-dependent RNA polymerase that's made of RNA, is about 40 nucleotides. And it turns out that the RNA you need to do this is about 250 nucleotides long. Yes?
So, from this theory, it's that RNA as a molecule in and of itself was the first organism of life, or that there were like cells formed that used RNA as their genetic material.
So the question is here, sorry, I'm going to paraphrase your question as well, but basically, do people think of this autocatalytic system as being life, or was there something else in terms of life above and beyond the RNA? And so most people, again, think about life in terms of a cellular life as opposed to a molecular life. But again, we can argue, and this is kind of hair splitting as far as it's concerned. But the idea that most of these people have is you have to have some kind of self-sustaining molecular system before you can have a cellular system. But again, people argue about that too. Is this one of the great things about talking about things 3.5 billion years ago is nobody knows. So you can argue basically till you're blue in the face. And some people do. But the idea here, at least as far as this class is concerned, because there are some connections, is that at least on a molecular level, you have to have something like this. You have to have a system where you can make more of that same system, and also have a system under which you can go evolution. Because if you just have a system where it makes exactly the same thing all the time, then you're not going to evolve. You're not going to have anything which progresses. And so in that way, RNA also makes a lot of sense. And we have a bunch of currently functional ribozymes. We talked about the ribosome already. Self-splicing introns we talked about already. One of the ones we haven't talked about so much is RNase P. RNase P is the ribonucleoprotein complex now, which cleaves off the 5' end of tRNAs. And it turns out that just the RNA from RNase P can do that. Most of the time it's got some extra proteins involved. Again, a lot like the ribosome. The ribosome has a bunch of proteins involved too. It seems to be much better with these proteins. But the RNA, just by itself, can do this. There are, however, a couple of problems with this idea that RNA could have been the first molecular self-replicating system. First, ribose is not a very stable molecule. And if you think about what this planet was like 3.5, 4 billion years ago, there wasn't a moon to start with, and then there was a really big asteroid that hit the Earth and blasted the moon off. It probably wasn't a real happy place to be during that period of time, what the people call the major bombardment. And so having a not terribly stable molecule that you're basing everything on at a molecular level might not be such a good idea. It turns out that not just ribose is not terribly stable. RNA, we already talked about being not terribly stable because of that 2' OH, but not just because of the 2' OH. As long as you have a 2' OH and phosphodiester formation, you're very unstable. And so a lot of people are trying to come up with ideas on how you could have had a genetic system and potentially also catalytic activity without having to have ribose like you have here. So some people talk about peptide RNA. So now you have these you have peptide bonds here, or pentose RNA. So this is your, sorry, on this side is the peptide nucleic acid. Peptide bonds are way more stable than phosphodiester bonds. Pentose RNAs, instead of having, sorry, this should be pentoses here. These are hexose RNAs that might be a little bit better off. So just in terms of the backbone, you can talk about stability. But on the other hand, we do know that there are lots of different structures that you can form in RNA in order to get some stability, but not so much stability. It's more in terms of giving yourself the appropriate chemical groups so you can have catalysis. We've already talked about double-stranded RNA, hairpin loop structures, but there's a whole bunch of different kinds of RNA structures which you can find. One of my favorites is this one down here, the so-called pseudoknot. It's just a single piece of RNA from 5' to 3', but has two separate base pairing interactions. And unfortunately, I don't have a three-dimensional structure to look at this, but it's basically these are two continuous helices with these two loops that come out on either side. You find these very often in viral genomes where you have translational frame shifting that takes place. So we talked about translational frame shifting last time. It's how the ribosome gets moved back or forward one nucleotide or sometimes two nucleotides because of a secondary structure which is formed in the RNA. For things like HIV, turns out it's exactly this structure right here that you have. Most of the rest of these structures here have been found in either tRNAs or in the ribosomal RNA, where you have all of these different structures which are forming in your RNA. Because of all these different structures, not surprisingly, you can generate different functions. Yeah?
The structures that are found in tRNA and ribosomal RNA, are they usually associated with a particular function? Or do they just sort of, they're observed and it's not clear if they sort of...
Yeah, so the question is, do people have a real direct connection between these structures and particular functions? And the answer is, in a few cases, yes. In many cases, no. Probably what most of these structures are here has to do with interactions of the different parts of the RNAs. So if you think about the large subunit of the ribosome, it's mostly one big RNA. And that has to have a particular structure, again, for the large subunit, where you're going to be catalyzing peptide bond formation. But holding everything in the right place in order to do that is probably what's giving you that particular function. So your individual bases, which are going to give you that activity, are going to be relatively small. But you've got a whole bunch of extra stuff which is giving you that structure. And that's exactly what you have in proteins, too. In terms of proteins, you have an active site, which is just a couple of amino acids in a big protein chain. And so it's probably very, very similar in terms of those kinds of functions. So not, one, you have a pseudonode, and that pseudonode is just giving you particular activity. It's all of these things together are making something which, in the case of the ribosome, gives you peptidyl transferase activity. So again, lots of interactions, probably mostly structural, and a few of them are going to then be enzymatic in terms of their function. Probably the best understood of these is basically something which, again, we've talked about quite a bit already. If you think about splicing, what is splicing? It's cutting RNA and religating RNA. What does most of that job? RNA. So it's not terribly surprising that you have, RNAs which have both endonuclease activity and ligase activity for RNA. The best studied of these is what's called the hammerhead ribozyme. Why is it called the hammerhead ribozyme? Because it kind of looks like a hammerhead when you look at its structure. And it binds to specific RNA sequences through base pairing interactions and catalyzes the hydrolysis of that backbone and can recycle here. And so you've got ribozyme binds to RNA, cuts it, recycles, goes back, and this whole cycle can go through multiple times. And this is kind of how you define an enzyme, is that it can undergo multiple reactions. It catalyzes multiple different reactions. And so that is why a number of people argue that self-splicing introns are not really ribozymes. Why? Because once you've spliced yourself out, you can't go and splice another intron. And so it's only a single turnover enzyme, what people would say. But curiously enough, this is what one of the guys got the Nobel Prize for, was self-splicing introns, that they were RNAs which had enzymatic activity. Again, other people argue about whether it's really an enzyme at all. Whereas something like this, the hammerhead ribozyme, is clearly an enzyme, because it does undergo these multiple cycles. So this particular ribozyme is found in viruses, of course, the coolest things ever. Although these are really interesting because they're viroids. They don't even have capsids on the outside, and so again, we could argue on whether those are viruses or, at all or not. But none of these, either cleavage or ligation, is going to get you to that molecular self-replicating system that we talked about that we really need. So the way to do that is you actually have to have a RNA-dependent RNA polymerase that's made of RNA. So how do you do that? Because we don't have that. What are any of the RNA polymerases we've talked about so far? What are they made of? The RNA polymerases. Any of the RNA polymerases that we've talked about, do any of them have any RNA that's associated with them? So there's a reverse transcriptase which uses RNA as a template, but is it involved in the activity in terms of active site? No. Enzymatically, all of the RNA polymerases that we have are all protein. So bacterial DNA-dependent RNA polymerase, eukaryotic DNA-dependent RNA polymerase, the reverse transcriptase has the RNA in it, but that's only because it's serving as a template. It doesn't seem to have anything to do with the activity at all. So at least now, all RNA is being made by protein. And so does get you kind of a chicken and egg question. Okay, all of the RNA is made by protein, but the RNA is making the protein. How do we deal with this? So what people have done is they've tried to figure out how a RNA-dependent RNA polymerase made of RNA could have happened. Not this is the way it did happen, but how it could have happened. And this gets us to in vitro selection, which is what most of the RNA World origins of life people do. And again, it's what Dr. Lehman does in the chemistry department. Basically, what you do is you take a large pool or a large selection of nucleic acids with completely different sequences relative to each other. So you get to try as big a random set of sequences as you possibly can get. This is easiest to do with DNA. You make lots of random DNA molecules. You transcribe them into RNA using a protein, RNA polymerase. And then these then are multiple different RNAs that can have multiple different structures. And then what you need to do is the needle in the haystack approach. Is you need to find one of these that has the particular activity that you're looking for, say, a RNA-dependent RNA polymerase activity. This particular example is actually looking at ATP addition. So it's basically, it's a kinase which you have then with an RNA activity, and it adds this phosphate to an RNA. However, the way this is done in this particular experiment is you actually included a sulfur residue in your ATP. It's ATP gamma S. It's that gamma is that last phosphate which you have in your triphosphate. And then you find the one RNA that has this phosphate group attached to it with the sulfur that gets separated from all of these other RNAs. It turns out this is the orange RNA here. And then what you do is normally you'll take this RNA, which is literally in some cases 1 in a billion or 1 in way more than a billion, and then make DNA copies of this using reverse transcriptase, and then do the same process again and again and again. So lots and lots of cycles. And eventually, you'll end up with a number of RNAs that have this activity, have this kinase activity. And then you sequence these RNAs, and you can then actually make those specific RNAs and show that they have this kinase activity. But the key here is you have many, many, many different RNA sequences which can form lots of different structures, some of which may have the activity that you're looking for. And the key here is to having a really nice selection for finding the one that you're looking for. And in this process, we found RNAs that have RNA polymerization activity, so RNA-dependent RNA polymerase, but again, only up to about 40 nucleotides so far. And the RNA that you need to do that is a couple of hundred. Also DNA polymerization, et cetera, et cetera, et cetera. And most exciting, at least as far as I'm concerned, is some of the ligases. You don't see that in here, but we'll take a look at one of these ligases, RNA ligation up here. Certainly in vitro selected RNAs, but RNA ligation, which you already have in the self-splicing introns, as they also have RNA ligation. So what do you have to have? Again, an RNA replicase. I haven't updated this. We're actually up to about 40 bases that this can do. The other thing you need to have, and getting back to your question, is cellular life, right? How do you get cells? Because all life that we know of now is cellular life. How do you get that separation, the compartmentalization taking place? And so there are a number of researchers that are looking into this, particularly Jack Shostak at Yale, who are looking at some of the potential prebiotic molecules, and some really cool experiments that people are doing with meteorites. So what do you find on a meteorite? And then on that meteorite, could any of the molecules you find there actually form some kind of compartment? And it looks like some of them could, which is actually really pretty cool. And then Eugene Koonin, is one of my colleagues, thinks that there could be some inorganic compartments which form in things like hydrothermal vents or hot springs that could also provide a compartmentalization that you could then maybe have your RNAs in separate from the rest of the world. And we talked about ligation here. This is, oops, hang on, it's missing my, oh, okay. So recombination, this is what Dr. Lehman does, and he had an article in Nature a couple of years ago, which is in my PowerPoint slides, but it's not showing up here for reasons that are not obvious. But so that's the idea for the RNA world, is that RNA, self-catalytic, and has lots of different activities, could have been the molecular origins of life, but again, we don't know if that's the case. If that, however, were, we could have this molecular system here, which is just based on RNA. We know, of course, that the ribosome is RNA. So RNA could be important for going to protein, but what about DNA? How do you get DNA? We've got, of course, all of our cells are DNA. All other cellular life has DNA for its genetic material. How do you get from RNA to DNA? And sorry, this should be RNA to DNA here. So RNA to DNA, turns out you just need two activities. You need reverse transcriptase, right? Because you take an RNA copy and make it into DNA. We already talked about the retrotransposons. We talked about telomeres. All of these are reverse transcriptases. And you also need a ribonucleotide reductase. You need to go from RNA to DNA. So ribose to deoxyribose. Turns out that most of the diversity that you find in ribonucleotide reductases and reverse transcriptases you find in viruses. Exactly. So viruses invented DNA. Okay. It's kind of a stretch, but there are some actually really pretty reasonable arguments about how viruses could have invented DNA. The first one is that viruses outnumber everything else on the planet by a whole bunch, probably 1 to 2 orders of magnitude. Some of the last calculations are that you've got 10 to the 30th to 10 to the 31 viruses on Earth alone. That's an insane number, absolutely insane number. If you look at the genome sequences that you find in these viruses, they're way more diverse than anything else. Many viruses have reverse transcriptases and ribonucleotide reductases, and it turns out the diversity in each of those sequences is much higher than you find in cellular ribonucleotide reductases and reverse transcriptases. And if you think way back to lecture 1, we talked about the diversity that you find in different organisms from different domains. The more diverse they are, the more likely they are that they're old. Well, okay, they're more diverse in viruses, so maybe they're older. And many of these virus proteins do not look like any kind of cellular proteins. So with that review of our RNA world, all you really need is RNA, at least in theory. We haven't completely shown that's the case. Something that looks like it. These could be your peptide nucleic acids or your hexose nucleic acids. Ribosomes are pretty good. They're not great yet, and the last part here is obvious. And small RNAs are really where it's at. This was the cover of Science a couple of years ago. So we'll see you all on Wednesday. Wednesday is review. There will be 2 clicker questions, and if I decide to put them online so you don't have to show up, I will post that on D2L.